Good morning, friends. In this video tutorial, we will be going to see about allosteric inhibition, a very essential topic in biochemistry. So, without wasting any further time, we will move on to the topic. So, what is allosteric inhibition? It is actually a regulatory mechanism. So, allosteric inhibition is a regulatory mechanism which regulate the activity of enzymes which regulate the activity of enzymes. So, we should know about it because the activity of the enzyme should be regulated during the biochemical process. Unless or otherwise, the end product will not be suitable for further production of other metabolites, right? So, to regulate the enzyme activity, there is an inbuilt mechanism in every cell called allosteric inhibition that regulates the activity of all the enzymes in the body, right? So, you should remember this point very clearly. Allosteric inhibition is a regulatory mechanism, right? So, in this mechanism, a molecule called regulatory molecule bind to a specific site on an enzyme. So, as we all know that, for example, let me draw a small diagram, right? Imagine this as a enzyme, right? This as a enzyme, right? So, this site is called as active site, and this is called allosteric site. Allosteric site. This is called allosteric site, right? So, apart from active site, there is a place or there is a sequence or there is a region in every enzyme called allosteric site. So, remember this point also, right? So, the regulatory mechanism, a molecule that binds to the allosteric site, not on the active site. Okay. So, you should remember these points very clearly. So, how can you explain this mechanism? So, allosteric inhibition is a regulatory mechanism. Both in biochemistry as well as in biology, it plays a significant role, right? Let me explain in a little detailed way, right? So, as I told you, imagine this as an enzyme, right? This as an enzyme. So, here comes an what's that? A molecule called a regulatory molecule, right? So, to draw the regulatory molecule, I should take another color, right? And this regulatory molecule has high affinity towards the regulatory site, the regulatory site, right? So, as a result of this affinity, what would happen? This allosteric molecule will go and bind to this area, will go and bind to this area. Which area? Tell me. Yes, allosteric site. So, as a result of this binding, what would happen? So, we will discuss about this in the later slides, right? Allosteric site. So, this is what we saw in the previous slide, okay? So, apart from active site, this is called active site. So, active site is a place where the substrate binds. So, apart from active site, there is another site called allosteric site, right? This is called the allosteric site. This is called a allosteric site or regulatory site, right? So, this is the place where the inhibitory molecule will go and bind, right? Fine. So here comes an important point. So allosteric inhibition occurs at a site on the enzyme or receptor. Enzyme on the receptor. Second thing, allosteric site is different from the active site, right? Active site is the place where the substrate molecule binds, right? And allosteric site can be located some distance away from the active site. From this presentation, it is very clear that there are two distinct sites in an enzyme. One is active site, another one is allosteric site. And there is a definite distance between the active site and the allosteric site, right? Remember. Regulatory molecule. Regulatory molecule. What is a regulatory molecule? A regulatory molecule, often called an allosteric inhibitor. Allosteric inhibitor. So, what is the nature of this allosteric molecule? This will bind to the allosteric site, right? This will bind to the allosteric site. Another thing, this binding can be reversible or irreversible, right? So, while writing in your examination, try to emphasize these points, right? What is allosteric inhibition? Allosteric inhibition is a regulatory mechanism, regulatory mechanism, right? In both biology as well as biochemistry. So, it regulate so many metabolic process, so many metabolic process. And this regulatory molecule binding with the enzyme is reversible or irreversible, right? Okay, we'll move on to the next slide. So, what would be the outcome if an allosteric molecule or inhibitory molecule binds to the allosteric site? What would be the outcome? What would be the result? See, in the classroom, you are studying, you are attending slip test, you are attending multiple quiz, right? What would be the outcome of these assignments, right? To acquire the required CIA, right? Continuous internal assessment marks, right? So you are working towards an outcome. In the same way, what would be the outcome if the allosteric molecule or inhibitory molecule binds to the allosteric site? The key answer for this question is conformational change. It causes conformational change. Conformational change where? In the enzyme. In the enzyme. So, as a result of the binding of the allosteric molecule, there would be a conformational change in the active site. There would be a conformational change in the active site of the enzyme. So, as we discussed earlier, active site is the place where the substrate binds. Substrate binds. Remember this point. Okay. Now let me give a small representation. Imagine this as an active site. This as an active site, right? This as an active site of the enzyme, right? This as an active site of the enzyme, right? Remember, right? And now let me take another color. And here is the place where the allosteric substrate binds. Allosteric substrate binds, or allosteric molecule binds. So, as a result of this binding, there would be a conformational change. There would be a conformational change. So, as a result of this change, as a result of this conformational change, right, the change, the change in the active site conformation, the change in the active site conformation. So, as a result, right, remember the substrate molecules, the shape and the structure of the active, substrate molecules like this, but the active site is somewhat different. So there is a incompatibility between the shape and size of the substrate and the active site. So this will prevent the binding of the substrate molecule with the active site. Right? What brings the change in the conformation of the active site? The answer is yes, you are right. As a result of binding of the allosteric substance, right, brought the change in the brought conformational change in the active site. As a result, the substrate will not be able to associate or will not be able to bound to the enzyme active site. You all know that for the formation of product, what is essential? Yes, enzyme, enzyme plus substrate complex is essential. Enzyme substrate complex is essential because as a result of this complex alone, the product will be synthesized. The product will be synthesized. Right. Okay. This is an important slide. As a result of this binding, there is an inhibition of the enzyme activity. So here comes the major point, right? Conformational change. Conformational change often results in the enzyme becoming less active or completely inactive. So the conformational change prevents the substrate from binding with the enzyme. Binding with the enzyme. So as a result of this, it alter the enzyme activity or enzyme ability to bind to the substrate or reduce the enzyme's catalytic activity. Right? Allosteric molecule alter the ability of the substrate to bind with the enzyme. Since there is no ES complex formation, right? Since there is no EX complex formation, there is no product at all. There is no product at all, right? And this is a non-competitive inhibition actually, right? It is a non-competitive inhibition. So it's a form of non-competitive inhibition because the regulatory molecule does not compete with the substrate, right? There is no competition between the regulatory molecule and substrate molecule. As a result, this allosteric inhibition is categorized as a non-competitive inhibition. Non-competitive inhibition. Cooperativity, another important property. So, allosteric inhibition can exhibit cooperativity where the binding of one regulatory molecule at an allosteric site can affect the binding of additional molecules. Once allosteric molecules strongly bound to the enzyme, there won't be there won't be any binding of additional molecules in the enzyme. There won't be any further addition at all. There won't be any further addition. Reversibility. Allosteric inhibition can be reversible. So this point we already discussed even in the very first slide itself, right? The condition can be reversible or irreversible. That is based on the reaction status. It is based on the reaction status. So what is the biological significance of this reaction? This is very important. As we discussed, this allosteric inhibition has an impact on both biology as well as biochemistry. It regulates the activity of both the enzymes involved in major metabolic processes. Major metabolic process. In other words, it may be called as feedback inhibition. Feedback inhibition, right? So, allosteric inhibition is a non-competitive process but it has biological significance. Biological significance, right? Uh, so, what actually happens in the process as a result of the conformational change? So, this is how the active site remains in the enzyme. As a result of binding of the allosteric molecule in the enzyme molecule, right? The shape of the active site differs. How it differs? It differs like this. It differs like this, right? It differs like this. Hence, imagine this is how the this is what the shape of actives, substrate molecules, right? So there is an incompatibility. If a active site be like this, if an active site be like this, then it is easy for the product to bind here, right? There is a compatibility, but due to the binding of this allosteric substance in the regulatory site, there is a great change in the conformation of the active site. So this causes an incompatibility, thereby the enzyme will not bind to the substrate, thereby the inhibition is uh, prominent, the reaction will not proceed further. So this can be explained with the help of an enzyme called ribonucleotide reductase. Ribonucleotide reductase, a key enzyme involved in the synthesis of DNA, right? Ribonucleotide reductase plays a major role in the conversion of ribonucleotides RN ribonucleotides ribonucleotides into deoxyribonucleotides. Deoxyribonucleotides. So for this enzyme, for ribonucleotide reductase, ATP is the inhibitor. ATP is the inhibitor. If ATP binds to the when this ATP concentration is more in the cell, if the concentration of deoxyriboadenosine trinucleotide phosphate is increased in the cell, right, it means the concentration of ATP is also increased in the cell. So, as a result of binding of the ATP with the ribonucleotide reductase, this will not bind to ribonucleotides, and hence the enzyme of nucleotide synthesis will be blocked. This is a very good example for allosteric inhibition, right? Thanks for watching. Don't forget to subscribe. Stay tuned with me. Bye bye.